Nazilah from Non-Destructive Biomedical and Pharmaceutical Research Center, iPROMISE UiTM. So as per stated in the title, we use acid hydrolysis, heat moisture treatments, and combinations of these two treatments to produce resistant starch from corn and examine its applicability to be used as oral colon specific drug carrier for cancer therapeutics. Starch has growing interest in medical and pharmaceutical fields, and we choose resistant starch since it gives benefits to colonic drug delivery and also colon health because it can resist digestion by alpha amylase and B ferments into short fatty acids, and this can help in maintain colon homeostasis. Here, first native cornstarch hydrolyzed in 0.2 molar HCl buffer pH 2.2 to mimic the gastric simulations at 37 degrees C for 2 hours. And the dry product from this treatment, part of them will undergo into another treatment, which is heat moisture treatment. And we use at three different temperatures, 90, 100, and 110 degrees C at 2 hours and 16 hours separately. Another sample, we just directly introduce into heat moisture treatment at the same condition. So at the end of the process, we'll have three samples. The first one, acid treatment. Second, acid treatment undergo heat moisture treatment, which is the combination method. And the third one, as a heat moisture treatment, starch. So all this treated starch will undergo analysis using FTIR, DSC, SRD, and molecular weight. This table shows the physicochemical properties of starches, and it's stated here that heat moisture treated starch at 100 degrees C for 2 hours exhibit the highest resistance level amongst other samples with reference to the intra or intermolecular bonding. So if you look at the FTIR data, this value here indicate that it has higher or stronger intermolecular hydrogen bonds compared to other samples. And this is true because when we analyze using DSC, it showed that the value of native starch undergo heat moisture treated has higher values, which indicates it needs excess energy to break these strong hydrogen bonds. And from the SRD analysis, the percent crystallinity of this sample is lower compared to these two samples, which indicates that part of the amylopectin had been converted into the amylose. And this is one of the criteria in producing resistant starch. And this is also true when we analyze using molecular weight analysis, because the sample here produced less or lower molecular weight compared to the native cornstarch and the acid-treated starch. From these two images, we can see that both variants had similar morphology, which indicate that heat moisture treatment did not alter the morphology of the starch. And here, the tables represent the physical properties of these two starches at nanoparticles size. Okay, even though the resistant starch nanoparticles exhibit a comparable drug release as those of the native starch in the simulated gastrointestinal media, but we still believe that the higher amount of amylose in resistant starch favors the covalent conjugation with targeting ligand and drug in oral colon specific drug delivery system design, and hence it's still the potential candidate for a drug carrier.